Okay, let's unpack this. Have you ever noticed how traits, hair color, facial features, things like that, seem to run in families?
Mm.
Well, it's not just us humans. Plants have their own uh, really fascinating family trees. You can see in their flower shapes, seed colors, even how tough they are in the cold.
Yeah, definitely.
Today, we're taking a deep dive into strategies of inheritance from Brian Capon's Botany for Gardeners. We want to uncover the molecular secrets, really, behind how plants pass on their characteristics and what that actually means for us gardeners.
And what's so fascinating, I think, is that the very same systems of heredity that shape us, they also define our plants. Everything from a huge oak tree down to the tiniest moss.
Right.
So our mission here really is to understand the core principles, plant genetics, reproduction, connecting those invisible cellular things to the vibrant life and growth you actually see in your garden.
So moving beyond just knowing how to grow them.
Exactly. Getting to the fundamental why, you'll get insights that really transform how you look at your plants.
Okay, so we all see the results in the garden,
Mhm.
right? The specific shade of a rose, how tall a sunflower gets, or maybe how well a particular plant handles a cold snap. But what's really driving those characteristics down at the core?
Well, fundamentally, these traits are determined by specific genes or sometimes sets of genes working together.
Okay.
You can think of genes as sort of molecular instructions. They're carried on these structures called chromosomes, tucked away inside the nucleus of every single cell.
Uh-huh.
And it's these genetic blueprints that get passed down from one generation to the next through reproduction.
Right.
In plants, you can often trace these family lines just by looking. Similarities in flower form, seed color, maybe even, like you said, their hardiness against cold or drought.
Mm-hmm.
And when genes get mixed up through sexual reproduction, the offspring, well, they show recognizable family traits, sure, but also their own unique qualities.
Like siblings in a family.
Exactly like siblings. You see it in
Mhm.
human families, and you see it just as clearly if you grow a batch of seeds from one plant. No two seedlings are ever exactly alike.
And the person who first really figured out the basics of this whole system, this genetic blueprint, was actually a gardener, wasn't he? Someone with a keen eye for plants.
He absolutely was. The fundamental laws of genetics, the ones we still use today, were first recognized by Gregor Mendel. He was an Austrian monk, and he did this incredibly precise, patient work, hybridizing common peas in his monastery garden.
Peas. It always comes back to peas.
It does. His meticulous observations of how traits were passed down, these repeated patterns he saw, they were just so groundbreaking. They completely revolutionized biology and actually gave some really crucial theoretical backup for Darwin's ideas about natural selection.
Wow.
You have to imagine the patience, right?
Yeah.
Tracking things like pod color, seed shape, over generation after generation of peas, noting every little difference. That's real gardener's insight.
Definitely. So, okay, we've talked before about how plants grow, making new cells almost like a biological photocopier.
Mm-hmm.
But how do they create the next generation? How do they pass on that mix of family resemblance and unique traits?
Right. That's where two really critical cellular processes come into play. Mitosis and meiosis.
Okay.
Mitosis, like you mentioned, is that cell replication process for growth. Basically, making more cells so the plant can get bigger. Each new cell gets an exact copy of the parent cell's nucleus, all its chromosomes, all its genes.
The photocopy analogy.
Exactly, like making a perfect copy of the entire instruction manual, every page identical for the new cell. That's how a tiny seedling becomes a huge tree.
Okay, so that's mitosis. Then there's meiosis. Sounds similar, but you said it's different.
It is fundamentally different in its outcome. While mitosis duplicates chromosomes, meiosis actually reduces the chromosome number by exactly 1/2.
Halves it. Why?
Think about it. Most cells in a plant or in you have two full sets of instructions, two sets of chromosomes, one set inherited from each parent. We call that state diploid, or toucan for short.
Right.
When that diploid cell divides by meiosis, it doesn't make more diploid cells. It produces cells that have only half the number of chromosomes, just one complete set. We call that haploid, or N.
Okay, so it's like splitting the instruction manual perfectly in half.
Precisely. Each resulting cell gets just one complete set of instructions, and this halving is essential for sexual reproduction. It's why your body cells and the cells in a plant's roots, stems, and leaves normally have chromosomes in pairs. One set came from the sperm, one from the egg, combining to make that diploid organism. Meiosis has to happen to make those sperm and eggs, or their equivalents, in the first place.
So plants use this reduction, this meiosis, for something specific in their life cycle, to keep things going.
Yes, exactly. And here's a key difference between plants and animals. Animals primarily use meiosis to produce gametes, the sex cells, like sperm and egg.
Uh-huh.
Most plants, however, use meiosis to form something called spores. And these spores play this really crucial and sometimes quite surprising role in their reproductive cycles. It leads to this fascinating pattern called alternation of generations.
Alternation of generations. Okay, that
Cool.
sounds complex. Does that mean the plant we actually see isn't always the main part of its life? Or there's more going on than we realize?
Precisely. You've got it. This pattern, alternation of generations, it's actually the most common way plants reproduce. It involves the development, one after the other, of two distinct forms, or generations. A haploid one, called the gametophyte, and a diploid one, the sporophyte.
Okay.
And how these two generations look and which one is dominant has changed dramatically over the course of plant evolution. Let's look at a few examples.
Yeah, let's break it down.
Okay, think about mosses first. You know those dense, green, velvety cushions you see in damp, shady spots?
Sure.
What you're looking at there is the dominant generation. The tiny, green, haploid gametophyte. That's the main moss plant. This little green plant produces the sperm and eggs.
Right on the moss itself.
Right on the moss itself, often on separate male and female plants. Then maybe a raindrop splashes, carrying the sperm to a female plant, and fertilization happens.
Right.
The new plant that grows from that fertilized egg is the diploid sporophyte. It looks totally different, usually like a slender, brownish stalk with a tiny capsule on top.
Oh, I think I've seen those. Sticking up out of the moss.
Exactly. And it actually grows right out of the green female gametophyte, drawing nutrients from it. Its entire job, the sporophyte's job, is to produce haploid spores inside that capsule using meiosis, that reduction division we talked about.
Oh.
Those spores get released, float away on the air, and if they land somewhere suitable, they germinate and grow into new green gametophytes, starting the whole cycle over again.
Wow. Okay, so moss is mostly the haploid generation we see. What about ferns? They seem more complex.
They're more evolutionarily advanced. The elegant fern plant you recognize, with its big, often feathery fronds, that is the dominant diploid sporophyte.
So the opposite of mosses.
Right. Now, if you look really closely at the underside of a mature fern frond, you'll often see these tiny clusters, usually appearing as brown spots or stripes. Sometimes they're covered by a little flap of tissue.
Yeah, I've seen those little dots.
Those are clusters of spore capsules. Inside those capsules, meiosis happens, producing huge numbers of tiny, dust-like haploid spores.
Okay.
Now, if one of those spores lands on moist soil, it doesn't grow into a new, big fern right away. It grows into something completely different. A tiny, often heart-shaped, green plantlet, usually only about a quarter-inch across.
Huh?
That is the haploid gametophyte.
Wow, that's small. Does it even look like a fern?
Not at all. It's photosynthetic, makes its own food, and it produces both sperm and eggs on its underside. The sperm need a film of moisture, dew, or rain to swim over and fertilize an egg on the same or a nearby gametophyte.
Still needs water then.
Still needs water for that step, yes. Once fertilization happens, the new diploid sporophyte, the baby fern, starts to grow, initially attached to that tiny gametophyte. But it quickly grows much larger, develops roots and leaves, and becomes the big, recognizable fern plant, completely overshadowing its tiny, short-lived gametophyte parent.
Incredible. It's like a hidden phase of its life.
It really is. Now, fast forward to flowering plants, the angiosperms. Here, this alternation of generations has become even more specialized, and the gametophyte generation has become incredibly reduced.
So the plant we grow for flowers and fruit, that's the sporophyte.
That's the diploid sporophyte, yes, the dominant generation by far. The gametophytes, meanwhile, are now microscopic. They're completely dependent on the sporophyte for all their nutrition and protection.
Where are they then?
Well, the male gametophyte is essentially the pollen grain. Inside the pollen grain, after meiosis creates the initial haploid spore, a couple of cell divisions occur. You end up with basically two sperm cells packaged inside, ready for delivery. That whole pollen grain structure is the male gametophyte.
And the female.
The female gametophyte develops inside the ovules, which are tucked away inside the flower's ovary. After meiosis, usually only one haploid spore survives and develops into a tiny structure containing the egg cell. That's the female gametophyte.
So being microscopic is actually a big advantage for flowering plants. It lets them live everywhere.
Absolutely. It's a huge evolutionary leap. This dramatic reduction in size and the fact that they're protected inside the moist tissues of the sporophyte inside the flower within the ovule means flowering plants, and also conifers like pines are no longer tied to external water for fertilization.
The pollen tube delivers the sperm directly.
Exactly. The pollen grain lands, grows a tube down to the ovule, and delivers the sperm right to the egg's doorstep, so to speak. No swimming required. This allowed them to conquer and reproduce successfully in much drier habitats, which is a major reason they dominate most landscapes today, unlike mosses and ferns, which are still restricted largely to moist environments.
Okay, so we have meiosis shuffling the deck genetically, but if genes are just being copied and sorted, how do we get so much variation? Even siblings from the same two parent plants can look quite different. Why aren't they more uniform?
That's largely down to the sheer randomness of what happens during meiosis. It's the random segregation, the sorting of those chromosomes and the genes they carry.
Random sorting.
Yes. Remember, the organism inherited one set of chromosomes from each parent. During meiosis, when those paired chromosomes line up before dividing, it's completely random which chromosome from each pair ends up in a resulting spore or gamete.
Can you give us a simpler way to picture that? It sounds kind of chaotic.
Okay, imagine a really simple plant cell with just two pairs of chromosomes. Let's say pair 1 has chromosome A from mom and A prime from dad. Pair 2 has B from mom and B prime from dad.
Got it. Two pairs.
During meiosis, when these pairs split to form haploid cells, one cell might get A and B while another gets A prime and B prime. Or the first cell could get A and B prime and the other gets A prime and B.
So different combinations are possible just from those two pairs.
Yeah. Exactly. It's like slipping two coins. You can get heads, heads, tails, tails, heads, tails, or tails, heads. Completely random which combination you get in any single event. Now scale that up. Many flowering plants have, say, 20 pairs in chromosomes. That's 40 chromosomes total. The number of possible combinations of those chromosomes that can end up in a single pollen grain or egg cell becomes astronomical.
Wow, okay. I see
That's
that.
why you see so much variation even in seeds from the same parent plant. It's nature's genetic lottery, constantly creating new combinations.
And it's not just which chromosomes end up where, but also how the specific genes on those chromosomes behave, right? You hear about dominant and recessive genes.
Precisely. Genes don't all have the same influence. They generally express themselves in one of two main ways, dominant or recessive.
Okay.
When an individual inherits genes for two conflicting traits, say, one gene for tallness and one for shortness if one gene is dominant, its trait is the one you'll usually see. It masks the effect of the recessive gene.
Like tallness in peas or beans.
Exactly. Let's take beans. You might have pole beans, which grow tall, and bush beans, which are dwarf. Tallness, let's use a capital T for the gene, is often dominant over dwarfness, lowercase t.
So if a plant gets a T gene and a T gene, what happens?
Okay, let's trace it. If you cross a pure breeding tall plant, meaning it only has T genes, its genetic makeup is TT with a pure breeding dwarf plant, only T genes, so TT. All the offspring will inherit one T from the tall parent and one T from the dwarf parent. So their genetic makeup will be TT.
Okay, and they'll
all be tall. Because the dominant T gene masks the effect of the recessive T gene. We call this plant heterozygous for the height gene, meaning it has two different versions.
heterozygous all. Got it.
Now, here's where it gets interesting for gardeners and explains why traits can skip generations. If you cross two of these heterozygous tall plants, TT cross with TP.
What happens then?
Well, remember, meiosis randomly sorts the genes. Each T parent can produce gametes, pollen or eggs, carrying either a T or a T. So when they combine, you can get offspring with TT, TT, or TT combinations.
So the dwarf trait can come back.
Exactly. Statistically, you'd expect roughly a 3.1 ratio in the next generation.
Well,
3 tall plants, the TT and T1s, for every 1 dwarf plant, the T1s. That recessive T gene was hidden in the heterozygous parents, but it can reappear when 2 Ts meet up.
That explains those surprise dwarf beans from 2 tall parents.
It does. And this widespread presence of heterozygous gene combinations, hidden recessive genes, is the key reason why seeds from hybrid plants often don't grow true to type. They don't all look exactly like the parent plant.
Right.
So the practical takeaway for gardeners, if you want plants that are guaranteed genetic replicas of a parent, say, a specific rose variety or fruit tree cultivar, you can't rely on seeds from it if it's a hybrid. You need to use vegetative propagation.
Things like cuttings or grafting.
Exactly. Taking cuttings, grafting, division, layering these methods create clones, genetically identical copies of the parent plant. That's how horticulturists maintain those specific desirable traits in named cultivars you buy.
Okay, that makes a lot of sense. But we've talked about how precise mitosis and meiosis usually are. What happens when things go wrong, when there's a mistake in those cell divisions?
Well, nature isn't always perfect. As precise as these processes are, errors, or hiccups, as you put it, do happen. And sometimes these mistakes lead to really fascinating variations, some of which have turned out to be incredibly important in horticulture.
Like what?
Well, one type of error is called aneuploidy. This is where a cell and maybe the resulting plant ends up with either an extra chromosome, so instead of N or 2N, it might be N+1 or 2N+1, or it might be missing one, N1 or 2N1.
Does that cause problems?
Missing a whole chromosome is almost always lethal. The plant just can't develop properly. But having an extra chromosome can sometimes survive, and it might even lead to noticeable changes, like increased size in certain parts. There's a variety of Jimson weed called globe, which has broader leaves and rounder fruits that's due to an extra chromosome.
Interesting.
But a much more common and, frankly, much more significant type of chromosomal aberration in the plant world, especially for gardeners, is polyploidy.
Polyploidy. Okay, what's that?
Polyploidy means that the cells possess 3 or more complete sets of chromosomes. So instead of being diploid to 2, they might be triploid, 3N, tetraploid, 4N, hexaploid, 6N, even octoploid, 8N or higher.
3 or more whole sets. How common is that?
Incredibly common in plants. It's estimated that maybe a third, perhaps even more, of all flowering plant species are polyploids naturally. And a huge number of our cultivated plants, the ones we grow in gardens and farms, are polyploids. If you browse a plant catalog, you're seeing loads of polyploids.
Really? So what's the advantage? Why are they so popular in cultivation? What does polyploidy do for the plant and for the gardener?
Yeah, the big takeaway for gardeners is that polyploidy often results in bigger, better, stronger plants. That's the main reason they're so popular.
Bigger flowers, bigger fruit.
Often, yes. Compared to their normal diploid relatives, polyploids, especially tetraploids, 4N, frequently show increased vigor. They might have larger leaves, bigger flowers, larger fruits or seeds. They can also have increased food value in crops, produce more wood and timber species, or just have a generally larger, more robust stature.
And they might be tougher too.
They often are. They can show enhanced tolerance to environmental stresses, cold, drought, pests, diseases. This gives them a significant advantage, both in natural selection and, of course, in making them more reliable and productive for us gardeners.
Well, sounds like a win-win. Are there any downsides to having all these extra chromosome sets?
There can be. One common consequence of polyploidy, especially in triploids, 3N, is sterility. They often can't produce viable seeds. Because having an odd number of chromosome sets, like 3 in a triploid, really messes up meiosis. The chromosomes can't pair up properly before dividing, so you end up with genetically unbalanced spores or gametes. Tetraploids, 4N, can sometimes be fertile, but often polyploidy leads to sexual sterility.
So that explains why some fancy varieties don't produce seed or the seeds don't work.
Exactly, and it's why many popular polyploid cultivars have to be propagated vegetatively using things like bulbs, corms, rhizomes, tubers, or by taking cuttings and grafting. Think seedless watermelons or bananas, often triploids.
Right. Okay, so how do these polyploids even happen in the first place?
Well, it usually starts with an error during meiosis. Normally, meiosis produces haploid and sex cells, right?
Yeah, the reduction division.
But sometimes that reduction fails. The chromosomes double, but the cell doesn't divide properly, resulting in gametes, pollen or egg cells, that are diploid, 2N instead of haploid N.
An unreduced gamete.
Precisely. Now, if one of these abnormal diploid 2N gametes fuses with a normal haploid N gamete from another parent,
2N plus N gives 3N.
you got it. That results in a triploid, 3N plant. And as we said, these are often vigorous but sterile.
Okay. And tetraploids, 4N.
Tetraploid 4N plants can form if 2 of these unreduced diploid 2N games happen to fuse together. 2N plus 2N equals 4N.
Can that happen within the same species?
It can, yes. That's called autopolyploidy. But even more interesting, and perhaps more important in evolution and breeding, is when it happens between different, though usually closely related, species.
A hybrid, but with extra chromosomes.
Exactly. A diploid gamete from species A fuses with a diploid gamete from species B. The resulting tetraploid 4N plant is called an allopolyploid. Allo meaning other or different.
Allopolyploid. So these can actually break down the barriers between species. That sounds like a really big deal for creating novelty.
It is. It's a huge deal. Polyploidy is considered one of the main ways that new plant species form and new gene combinations arise in nature. It often gives the resulting plants significant advantages, allowing them to exploit new environments or outcompete their parents.
And we've harnessed this for crops and flowers.
Absolutely. This process, both natural and induced by breeders, has given us countless economically important and beautiful cultivars. Think about many varieties of apples, grapes, wheat, cotton, tobacco, potatoes. Also maize, rice, strawberries, and ornamentals like many roses, dahlias, chrysanthemums, gladioli, and even some orchids. Polyploidy played a key role in their development.
Wow. But wait, you said earlier that hybrids between different species are often sterile. How do these polyploids manage to be fertile and successful if they started as hybrids?
Excellent question. You're right, a simple hybrid between two different species, let's say species A crossed with species B, is often sterile. Why? Because the chromosomes from species A don't have matching partners among the chromosomes from species B. They can't pair up properly during meiosis.
So meiosis fails, no viable gametes.
Correct. But here's the magic trick of polyploidy. Sometimes, in that sterile hybrid plant, there's a spontaneous event where all the chromosomes just double. So now, instead of having one set of A chromosomes and one set of B chromosomes, the plant suddenly has two sets of A and two sets of B.
Ah. So now every chromosome does have an identical partner to pair up with during meiosis.
Precisely. Each A chromosome can pair with another A, and each B can pair with another B. Meiosis can now proceed normally, producing balanced gametes, each containing one set of A and one set of B, and suddenly this plant that was derived from a sterile hybrid becomes fully fertile.
That's ingenious. Nature finds a way.
It really does. The common garden dahlia, for example, is thought to be an interspecific octoploid hybrid, meaning it has 8 sets of chromosomes derived from different wild species, and it's highly fertile. This spontaneous doubling following hybridization is nature's workaround for hybrid sterility and a major engine for plant evolution and diversity.
Okay, so with all these discoveries about meiosis, random sorting, dominance, polyploidy, what's the next step? Where is plant breeding heading? Because relying on chance crosses and spontaneous chromosome doubling sounds a bit unpredictable.
It certainly can be. Traditional plant breeding, while incredibly successful over the centuries, does rely heavily on the chance outcomes of sexual reproduction. Breeders make crosses, hope for the best combination of genes from that random segregation we talked about, and then select the best offspring. They have very little direct control over which genes go where.
Right.
And even when they try to induce polyploidy artificially, maybe using chemicals like colchicine, the results are still pretty unpredictable. For a very long time, breeders were basically limited to working with the traits and genes already present within a single species, or maybe crossing closely related ones if they could overcome the sterility barriers.
But that landscape has changed dramatically now, hasn't it?
It absolutely has. With our deep understanding of DNA, the genetic code itself, and the development of really ingenious techniques for manipulating genes, plant breeding has entered a completely new era, the era of genetic engineering. It's now actually possible to isolate individual genes, the specific bits of DNA code for a desired trait, from one organism, maybe one plant, and splice them directly into the chromosomes of a completely different plant.
Not just mixing whole sets of chromosomes anymore, but picking individual instructions.
Exactly. We can target specific characteristics. And what's truly revolutionary is that these gene transfers aren't limited by the old species barriers. You can potentially move a gene from, say, a bacterium into a corn plant, or from a cold-hardy Arctic plant into a crop species, or even between completely unrelated plant families.
Wow, across kingdoms almost.
Yeah.
And do those inserted genes work properly?
Generally, yes. These inserted genes typically function normally within their new host cell and plant. They simply direct the cell's machinery to perform new biochemical tasks or express new traits, whatever that gene codes for. And a huge advantage is once you have a successfully engineered plant, you can often propagate it by cloning vegetative propagation again.
Bypassing the whole unpredictable sexual reproduction lottery.
Precisely. It offers unprecedented precision and efficiency compared to traditional methods.
So what kinds of custom-made plants could we realistically see in the future using this kind of precision genetic engineering?
Well, the possibilities theoretically seem almost limitless. You can imagine agricultural crops engineered for remarkable natural resistance to devastating pathogens or harmful insects, which could dramatically reduce our reliance on chemical pesticides.
That would be huge.
Or think about plants designed to thrive in challenging environments. crops that can tolerate drought or grow in salty soils, perhaps by borrowing genes from desert plants or salt marsh species that evolved those abilities millions of years ago.
Adapting crops to climate change, maybe?
Potentially. We could see plants with enhanced nutritional value, maybe boosting vitamins or essential amino acids,
Hmm.
crops with higher yields, plants producing stronger fibers for textiles, trees engineered to grow straighter wood for construction.
It sounds like plants could become factories.
They already are in some cases. Ordinary plants are being explored and used as living biofactories to produce complex pharmaceuticals, like vaccines or antibodies, potentially much more affordably than traditional methods. And because, theoretically, gene sharing knows no taxonomic bounds anymore, maybe even fundamental processes like nitrogen fixation.
Which only certain bacteria can do now.
Right. Maybe that ability could be engineered into major crops like wheat or rice, reducing the need for massive amounts of nitrogen fertilizer. The potential impacts on agriculture and sustainability are really profound. So when we bring it all back, this deep dive into plant inheritance, into meiosis, gene sorting, alternation of generations, polyploidy, it really shows how understanding these fundamental biological processes isn't just abstract science.
No, it connects directly back to the garden.
Exactly. It gives us practical knowledge that informs everything from our watering practices to how we prune, how we manage pests, and especially how we propagate plants. It helps us grasp the why behind the how of gardening.
So what does all this mean for you listening? Well, next time you're out in your garden, maybe take a closer look at your plants. See them with this new perspective. Every leaf, every flower, every single seed pod is this incredible testament to billions of years of evolution, these intricate genetic strategies playing out right there. It's a marvel of biological engineering in your own backyard.
Mm-hmm.
And as for that future, the custom-made plants, the possibility maybe someday of seeing a truly blue rose or bright red delphinium rolling off the genetic engineering assembly line, breaking all the old rules. Well, whether that's ultimately a good idea, that's something for you to think about. Thanks for joining us for this deep dive into the fascinating world of plant inheritance. We really hope this knowledge helps you appreciate the incredible biology at play in every single plant you encounter. Keep exploring, keep learning, and happy gardening.